All right, I'm going to go over the biology first semester final exam study guide. If you're just copying these answers and not really thinking about it, you're kind of wasting your time. All right, anyways, proteins. I know this one's on the test. What are they made out of? They're long strings of amino acids. Proteins are made of amino acids. I'm going to put down three functions, three jobs that proteins do. Going to need to know them. All right, so number one, they speed up chemical. I'm going to put RxTS for reactions because I can't fit it. They speed up chemical reactions. They carry oxygen in blood, and they also help to fight disease. All right, so what are the examples that do these three things? The things that speed up chemical reactions, the proteins, are called enzymes. The proteins that carry oxygen in blood, I misspelled it, it's a Z, is hemoglobin, globin. And the proteins that fight disease are antibodies. All right, so let me fix this. Enzymes speed up chemical reactions. Hemoglobin carries oxygen in blood, and antibodies fight disease. Carbohydrates, I don't think this is on the test, but they're made of sugars. Made of sugar. What's their job? Provide energy. And what's some examples? Let's do the ones from nucleic acid. Deoxyribose is the sugar in DNA. And whoa, I got to tap. And ribose is the sugar in RNA. Lipids, I don't think it's on the test, so I'll do it anyways. Fatty, they're made of fatty acids and glycerol. Functions, they make up most of the cell membrane. I'm not going to type that. And remember, phospholipids. That's the type of lipids that make up cell membranes, the outer covering around each of your cells. Nucleic acids, we should know this because we just finished this unit. They're made of nucleotides. So DNA and RNA are made of a bunch of nucleotides bonded together. Function, store genetic information. And examples, obviously, DNA and RNA. All right, moving on. All right, some of these I'm not going to type them to make this shorter. An enzyme is a protein that speeds up chemical reaction. Plus, I'll be able to see who's actually listening to me and who's just watching me. An enzyme is a protein that speeds up chemical reaction. All right, right here, I'm going to label this. This right here, this is the enzyme. Enzyme, the blue thing. Right here, this space right here, it's got the perfect shape that this molecule is going to fit in there. That's the active site right here, active site. And this molecule right here, this is the substrate. So this enzyme, the blue thing, is going to work on this substrate. It's going to turn the purple thing into this green thing and this yellow thing. And these things over here, that's the product. Product. All right, so remember, it's the shape of proteins, it's the shape of enzymes that determines their function. If their shape gets screwed up, they will no longer work. All right, moving on. What does it mean that enzymes have specificity? It means they are specific to one substrate. They only work on one substrate because they have a perfect shape that works with that substrate, doesn't work with anything else. What factors affect enzyme activity? You'll need to know this. I'm going to say this. Enzymes, they work within a specific pH and temperature range. So if the pH gets too high or too low, they'll stop working. If the temperature gets too hot, they'll stop working. This is just reading a graph. What is the optimal pH for the enzyme in green? Optimum is when it works the best. It's when it's the highest. Looks like about, I don't know, 2 point something. I'll just put 2.3. What's the optimum pH for the enzyme in blue? All right, this one it's good over here. Looks like around 7. So this one, the one in blue, works best around 7. This one works best around 2 or 3. What is the optimum temperature for the enzyme in orange right here? This one, it's the best between 10 and 20. Looks like about 15. I'm not going to type it. This one works the best right here between 30 and 40. Looks like about 37, 38. Put that right there. I'm going to label this. You need to know that the outside, the balloon part of cell membranes, that's the phosphate heads. Phosphate heads. The balloon part. Now, the inside of the cell membrane, the strings in here, those are the lipid tails. You're going to need to know this. Lipid tails. And the last thing I'm going to label, I'm like, this is cholesterol, this yellow thing, but I don't think you need to know that. You need to know this. What is that made out of? That's not a carbohydrate. That's going to be a protein. All right, so that's the makeup of a cell membrane. I know there's a question on that. All right, cell transport. Simple diffusion, does it require energy? No, because it's passive transport. Facilitated diffusion, does it require energy? No, because it's passive transport. A type of passive transport. Osmosis, does it require energy? No, because it's passive transport. Active transport, does it require energy? Yes. All right, which way do molecules move? Top three are going to be the same. It doesn't require energy because the molecules are moving from high to low concentration. Screwed it up. Let me put it over here. High to low concentration. Facilitated diffusion, same thing. Molecules are moving from high to low concentration. Osmosis, same thing. The water is moving from high to low concentration. The only one that's different, active transport, it requires energy because the molecules are moving from low to high concentration. That's why it requires energy. Facilitated diffusion requires the assistance or help of proteins. Channel protein, carrier protein needs a door to allow things in and out. Osmosis, whenever you see that word, water, whenever water is moving across the membrane, that is osmosis. What type of transport above are considered passive? The top three: diffusion, facilitated diffusion, and osmosis. I'm going to type it again. Passive transport does not use energy, and active transport does. All right, the little shapes here. Molecule A is the triangles, molecule B is the hearts, and the ovals are molecule C. All right, if molecule A is permeable to the cell membrane, that means it can go through it. Which way would it move in diffusion? All right, so we're talking about the triangles here. There's 6 of them in here, there's only 4 out there. So there's a high concentration of triangles inside the cell, low concentration outside the cell. So through diffusion, the triangles will move out from high to low concentration. Which way will molecule C moves? And is this diffusion, osmosis, or active transport? How do you know? So if it moves out of the cell, so out here you can see there's a bunch of these little circles, and there's only 3 inside. So there's a high concentration of the little circles out here, low concentration in here. If those circles are moving out from low to high concentration, we know that it's active transport. It's got to be active transport. And how do we know that? Because the substances, the circles are moving from low to high, and the only way things move that way is through active transport. All right, moving on. Osmosis. All right, so right here we have a cell or a balloon or something, and inside it's 45% water, outside it's 75% water. You guys are good at math. 75 is a bigger number than 45. So in this case, water would move in to this cell from high concentration, 75%, to lower concentration, 45%. The one over here, it's 90% water inside, it's 63% water outside. 90 is more than 63. So the water through osmosis is going to move out of the cell. All right, let's see. All right, I'm not going to type it because I just did it with arrows. Define homeostasis. You need to know this. All right, it is when an organism maintains a stable or balanced internal environment. So the conditions inside their body stays environment. I misspelled it. You can spell it right. All right. So the conditions inside their body stays stable, like the body temperature, blood glucose, things like that. It stays relatively stable. All right. What are 3 ways animals maintain homeostasis? I'm going to put 2. All right. Birds ruffle. I don't even know how to spell ruffle. I guess that's it. Feathers when it is cold. That helps them stay warm. How about bees? They like buzz. They buzz when it is cold to help them stay warm. All right, so that's 2 ways that animals maintain homeostasis in their body. All right, photosynthesis and respiration. I'm not going to write the equation because if I give you the reactants, the inputs, and the outputs, all you got to do is put the inputs on the left side of the arrow, put the outputs or products on the right side of the arrow. Photosynthesis, inputs. What do plants need? They need CO2. They need H2O, water, and they need light. What do they make? What are the outputs? They make glucose, sugar, and oxygen gas. Where does it occur? It occurs in the chloroplast. All right, cellular respiration. Once again, I'm not going to give the equation because just put these reactants or inputs on the left side of the arrow. Put the products or outputs on the right side of the arrow. All right, what does cellular respiration need? It needs glucose, sugar, and oxygen. And what does it make? It's going to make CO2. We breathe it out. H2O, water, and ATP. And where does it occur? Mitochondria. All right, the 3 steps of cellular respiration. I know in my class I had 4, but a lot of textbooks put step 2, pyruvate conversion, and step 3, Krebs cycle together. So it looks like that's what we're doing here. All right, so step 1 is glycolysis. How many ATPs? You won't have to know exactly, but you should know that it doesn't make much. It only makes 2. The second is called the Krebs slash citric acid cycle. How many does it make? You won't have to know exactly, but just know it doesn't make very much. It makes 2. And the last step is called the electron transport chain. You won't have to know exactly how many ATPs, but you should know it makes the most by far. It makes about 34. All right, why is ATP so important? That's simple. It can be used as an immediate source of energy. ATP is the energy molecule. All right, DNA and protein synthesis. All right, so here, this is a nucleotide right here. You got the base, the sugar, and a phosphate. So if there's an A over here, there's going to be a T or thymine over here. This right here, this shape, this is going to be, I'm just going to label it D for deoxyribose sugar. And this right here, that's the phosphate. I'll put P for phosphate. And obviously, if there's a G over here, there'll be a C over here. All right, base pairing rules is simple. If there's an A, there'll be a T. If there's an A on the left, there'll be a T on the other side. If there's a T on one side, there'll be an A because A goes with T, except RNA. T goes with A, except RNA because it goes with U. G goes with C. G goes with C. C goes with G. And C still goes with G. I'm not going to do transcription on this little strand right here because I'm going to do it. The next question asks me to do it, and so I'll do it just once. All right, transcription. Where does it take place? Since we're copying the code in DNA and DNA is in the nucleus, it's got to take place in the nucleus. What is made? We're copying the code of DNA and making a messenger or mRNA. What type of RNA are involved? So this is going to be M or messenger RNA. Going over here, translation, the second step of protein synthesis. Where does it take place? It's where proteins are made, ribosome. What is made is where we're making the protein. Protein synthesis literally means making proteins. What type of RNA are used? We're using rRNA, tRNA, remember they're carrying the amino acids, and the messenger RNA that has the code from DNA that knows how to make the protein. Messenger RNA, what's its abbreviation? We just said it, mRNA. What's its job? What's its function? Copies the code in DNA and takes it to ribosome. tRNA. Transfer RNA is also known as tRNA. What's its job? Carries amino acids to ribosome because we cannot make proteins without amino acids. And the least important in my class, ribosomal or rRNA. What's its job? Makes up the majority of ribosomes. All right. All right. Transcription, translation. So we got a segment of DNA here. I'm going to separate it. Remember, DNA and RNA talk in 3-letter words, not 1, not 2, not 4, but 3. So I'm going to show the 3 bases. All right, so now I'm going to turn this DNA into mRNA codons. So A will go with U because there's no T in RNA. T goes with A. G always goes with C. The next codon, G goes with C goes with G, A goes with U. The next one, T goes with A, T goes with A, C always goes with G. Next codon, G goes with C, G goes with C, A goes with U. Third, the next one, T goes with A, T goes with A goes with U. And the last one, C always goes with G, G goes with C, and G goes with C. All right, so what amino acids are going to show up? The first codon is UAC. We start in the middle. U, A, C, tyrosine. All right, so TYR is the first amino acid in the protein. The next one, CGU. Start in the middle. CGU, arginine. ARG. AAG, what amino acid is coded for by AAG? Let's see. AAG lysine, LYS. The next one is CCU. Start in the middle. CCU proline, PRO. AAU. Start in the middle. AAU asparagine, ASN. And the last one, GCC. GCC alanine, ALA. All right, so going from DNA to mRNA, that's transcription. Going from messenger RNA to the amino acids and making a protein, that is translation. All right, so the last question, and I believe this has to do with the written question on our test. I'll repeat that. I believe this has to do with the written question on our test. It says, what would happen if there was a mutation in the third codon? So right here, the third. So instead of it being TTC, the DNA makes a mistake. Whoops. Instead of putting a T, it puts an A. All right, so it made a mistake and put an A instead of a T. How's that going to affect the messenger RNA? Well, now it's not going to be A goes with A. It's not going to be an A anymore. Now it's going to be A goes with U, T goes with A, and C still goes with G. So it's changed the codon. The codon is no longer AAG, it's now UAG. So are we going to still have the same protein? Are we still going to have lysine as the third amino acid? Well, let's look. Now it's going to be UAG. So we start in the middle. G, that's going to be a stop codon. So it's no longer going to be lysine, it's going to be stop. It's a stop codon. So instead of the protein continue to be built, it's going to stop after tyrosine and arginine. So what does that mean? So the mistake, the mutation in DNA, it will totally change the shape and function of that protein. So changing the DNA changes the messenger RNA, which then can sometimes change the amino acid, which will change the protein. Because remember, it's the order of the amino acids that determines what shape the protein is going to have, and it's the shape of the protein that determines its function or job. All right, I hope you guys understood this. If you did, and you wrote all this down, you should do well on the test. All right, I'll talk to you next time.